Please welcome to the stage Dr. Josh Clevenger.
All right, am I on? I am on. They said it would be on when I got out here. So I have to start out by just saying what an absolute honor it is to be on this stage, talking with you at a PacBio event, because specific biosciences long-read sequencing has revolutionized translational genomics. And as a father, getting to hear about the amazing impacts on human health that HiFi is having and is going to have, and then listening to Jonas talk about how all of that happened in 22 years and thinking about my children and how old they'll be in 22 years and the next wave of innovation. It's just really incredible to be here and to talk to you. And my group specifically focuses on how we can translate genomics for direct real-world impact. So food security. So what that means is it means that if you think about all the smart people that care about the human genome and that work on insights in the human genome, my group alone in the last 5 years has had to understand insights in 65 different species, with genomes ranging from the size of 500 million base pairs to 22 billion base pairs, and ploidy levels 2, 4, 6, 8, and everything in between, sometimes 5, which is a little weird. So if you think about the amount of effort it takes to understand everything about the human genome and how HiFi has impacted that, you can start to understand my perspective for my goals, just how incredible HiFi sequencing has been. And it started as just sequencing a genome. So I was one of those groups at a large genome consortium that had given up on assembling a genome. After $6 million in 10 years, before long-read sequencing, the Peanut Genome Consortium said, we are never going to sequence and assemble a cultivated peanut genome. It was just too hard. In just the first round of long-read sequencing from PacBio, we were able to do it. And so all of a sudden, that's possible. And then all of a sudden, it became possible to sequence 50 peanut genomes and to understand pangenome graphs, and to understand all the variation between those individuals, all the things that we couldn't see before. Because we sequence tens of thousands of samples from plant species all around the world. And we work with groups, public and private, to try and make an impact on the problem that they're trying to solve. Whether that's identifying genes that control the plant's ability to resist disease or tolerate drought, or that's making predictions on how that plant will thrive in different environments. And so having the ability to see all those differences is a massive, massive step change. And in breeding, we talk about step change. We talk about that one line that's going to be better than every other line for the next 10 to 20 years, and there's nothing you can do about it. So when the Revio came out, I was at PAG, and a colleague said to me, you know what, Josh? It's really cool what you do and everything, how you use low-coverage sequencing at scale to do translational genomics. But now with the Revio, shouldn't you just do everything with long reads? And first I thought it was kind of a joke. I was like, okay. But then I thought about it. I was like, yes, we should do everything with long reads. And what was amazing about that is that it wasn't possible on that day, but PacBio kept pushing the envelope on accessibility and made it possible. And so the idea of having scaled long-read sequencing at the population level was something that we couldn't pass up trying to do. Because for every crop that has a threat associated with it, there's a child that relies on it for its life. And there's a grower and a farmer that relies on it for its livelihood. And so every year that we don't have a solution to that problem, all of those folks that rely on that crop have to suffer for it. There's just nothing else that we can do. And so the ability to get to tools faster and better than before was something we couldn't pass up. And so Veil Genomics is about the power of scale. Because just because something is possible doesn't mean it's accessible. So our value proposition is we want to bring scaled long-read sequencing. We want to make it accessible. And so my colleague Alex Harkis, who gave a great talk in a workshop earlier today, and I created Veil Genomics along with postdoc Kendall Lee, with the idea that we had the dream that every single group, every single person that wanted to use the power of long-read sequencing at population scale should have that ability. And that was our value proposition. So today what I want to do is I want to take you through some projects in our own labs that can show you how excited we are about what is actually possible with this. And if we start out, okay, we think about pangenome graphs. You just think of the first level of what HiFi has done for agricultural genomics. Here's a great example. This is in soybean. So this is high protein, low oil in soybean. And soybean is incredibly important around the world. And so you're talking about folks were trying to understand the locus of high protein in soybean for decades. Think about that. Across the world, groups trying to map this trait, spending millions of dollars and time and energy. Well, it turned out that if you use SNP arrays or GBS or some other technology, you never find it, because it's a 300 base pair deletion that controls this trait, and it has cryptic linkage disequilibrium with a SNP that's about 1 million base pairs away. So you would never find it. They did a massive resequencing study to identify that, yes, from the wild soybean, it was this deletion that caused high protein. And then my colleagues at the University of Georgia, who had been trying to map this trait, went back and they put a pangenome together and mapped it just like that. So imagine that impact, maybe a decade, maybe more, millions of dollars around the world, but if they had just had the ability to map on the pangenome graph, they would have found it immediately. That's pretty incredible. If we have the opportunity to do long-read sequencing scale, it ends up being a lot of small challenges that add up to one very large challenge. And that includes, we have to do extractions in a 96-well plate, so we're working with smaller and smaller amounts of tissue from loads of different species, which have different compounds in them, and it makes it really difficult. Then, after that, you have to size the DNA. Well, it turns out that mechanical shearing of DNA works really well across many different species. And so because of the work that PacBio did, it's actually possible in a 96-well plate to shear the DNA while you walk away and do something else and come back and you have perfectly sized DNA for library prep. And again, because of their drive to making their technology more accessible, they created library preparation that could be done in multiplexed and 96-well plate. There's another company called Sequel that produces a library preparation called longplex, which works very well also. And so there's multiple options for how you would actually take tissue all the way through to library preparation at scale. And I would be remiss if I didn't mention Ranger Lightbench, who is a sponsor in here. Their ability to do size selection and quantification of these libraries is actually really transformative as well. So there's a path forward for thinking about, I want to sequence 2,000 individuals, whatever species it is, and I want to be able to have and understand that data within a week or two. Now you can do that. So the next question is it better? We think it's better, we think the information is better, but is it actually better? And so we actually have the possibility to look at that in a project that I lead in southern Alabama. It's a breeding program where we only make selections with whole genome sequencing. So we do this in high schools. We go into the high school and the students take a peanut seed and they grow it and they take observations on its vigor and other phenotypes. We come in, we teach them how to extract DNA. We prep, sequence, and analyze that DNA with them. And then the students make the selection. They tell me, this is the selection that I want you to make. And then the next semester, the next set of students do the same thing. So they're learning about breeding, they're learning about how genomic technology advances breeding, and we're actually producing cultivars for the region. We sequence 300 to 500 peanut plants every semester. So we have a wealth of possibility here. So we took one of the populations and we said, what if we sequenced the entire population with long-read sequencing? How much better would it be if we had those data? The first thing that's interesting is that you want to know how much coverage do I need to understand having insights across the entire genome. So the peanut genome is a tetraploid. It's about 2.5 billion base pairs. And it turns out that at about 1x coverage, you're covering 70% of the gene space in the genome. And at 4x coverage, it basically plateaus. So we essentially only need to sequence that much to make an impact. So that was a good first start. The next place is, do I have physically more variation that I can see, right? That's the whole point of all this. And of course, the answer is yes, but you kind of have to show that as well. So if we take the long reads and we analyze them just on a linear reference genome, on the left is the amount of SNPs, and of course, we colored this for pink to represent long reads and the gray to represent short reads. The same samples, the same plants sequenced with different technologies. We get many, many more SNPs and indels. Indels are just the size smaller than a short read size, so about 150 base pairs or less, and then your larger structural variants. So we get a lot more. But what's really interesting, and I have to point out, and that these samples are all subsampled to be about 1x coverage, so it's the exact same amount of base pairs, right? So there's no difference in the yield. If we analyze them on a pangenome graph, right, because HiFi gave us, who care about peanut, the ability to have 50 genomes. So if we analyze them on a pangenome graph, it's exponentially better. So we're physically able to see more SNPs, more indels, and more structural variation. And a lot of that structural variation is making real impact on the phenotype of the plant. And we need to be able to see that. And so one of the questions that's pretty interesting is, if I have long reads at scale, do I need a pangenome graph at all? Well, it's about your taste, but in these samples, it looks like it's exponentially better to analyze them on the graph structure. So double HiFi, double wins for HiFi. That's really great, not super interesting. We kind of expected that to be the result. Well, what's next? Why don't I show you a real example? So here's an example of disease resistance in peanut. It's tomato spotted wilt virus disease. This is a disease that showed up in the 80s and was so disastrous that the peanut industry thought they'd never grow peanuts in the United States ever again. And so they found resistance in a line that was not, you wouldn't grow it. You wouldn't yield very well. The peanuts wouldn't taste very good. And they tried to map it to understand that resistance. Very strong resistance, major locus dominant, for 25 years. And in 25 years, they could never identify what was the underlying cause of that resistance. And it turns out it's a copy number variant. And it also turns out that you can have 2, 3, or 4 copies of this duplicated gene cassette. And the more copies you had, the more resistant you are, to the point where if you have 4 copies, you're able to resist essentially most of the viruses that affect peanut. So really transformative resistance that we couldn't see without HiFi. And now I'm going to show you that it's really actually hard to select for without HiFi. So if you want to put this in backgrounds in peanuts all over the globe, you have to be able to actually select for the causal variant that you're trying to select for. And that actually is really difficult. So let me show you how that looks like. First of all, when we look at lines, so we sequenced a few hundred lines with long reads. When you look at lines that you know have the insertion, and you look at it versus if you sequence it with short reads, coverage of the insertion with the long reads is almost 100% most of the time. And when you have the short reads, you basically hardly cover the insertion at all. Why is that? Because it's a duplicated gene cassette, and so there's lots of similarity between those genes within the duplication and also genes outside that are paralogs. And let me show you what that looks like. So this is sort of a mock-up of a line. And so you can imagine this across hundreds of lines happening all at once. With the long reads on the bottom, you see really nice coverage of the insertion. And this is real data that we sort of put to make it more accessible. But with the short reads, you never actually have any coverage over the insertion. And so you're never able to see that. And so you can't select for it, you don't know it's there, and it's essentially useless to you. And so this ribbon plot represents sort of what it looks like. Where the blue is filled in is actually where there's read coverage for this actual insertion, which is the bubble there. And so it's just a nice way to show that even at very low coverage, you're capturing all of that sequence and you're able to type that correctly and you have that really strong resistance. On the other side, because it's duplicated, you get all of this spurious mapping with short reads, and so you might say a line is resistant when it's not, which is also equally bad. You don't want to think something is resistant using genomics, and it turns out it's not. You put it out in the field and it dies. And so what that looks like is you actually get a lot of mapping in the insertion from the reads that are mapping there, but they actually should be mapping other places. And with the long read low pass, you actually have a lot of reads that span that gap, and you know for sure it doesn't have it. So I picked out two examples to show you what it looks like for those examples, but think about this happening over hundreds and thousands of individuals in informatics that take a few hours. And so you have the scaled sequencing. Once you get that sequencing, you have an answer within a few hours, and you can start to think about how impactful that can be in these breeding situations. So let me give you another example, which is really cool. So over the past decade in the Southeast, there's been these freezes that happen early in the spring that obliterate blueberry production. And what happens is, depending on where the flower and the fruit development is in the process, when that freeze happens, they can either withstand that freeze or die. And so breeders in the Southeast really want to try and understand the major loci where they can make selections for earlier fruiting or later fruiting, depending on how they want to do it. And so we have a colleague at the University of Georgia who has a very large population and wanted to understand these QTL so she can use them in her breeding program. And blueberry is a perfect example of a plant species where using genomic sequencing to make selections early on is incredibly impactful, because you can make thousands and thousands of crosses, you can have tons of progenies, but you're not going to know what the fruit are like or what the development looks like for 3 to 5 years. So if you could make those selections early on, you could skew your populations to the phenotypes that you want, and then you're just looking at other things like how good they taste, which in the end is what's most important. And so when we looked at this population of 1,000, and we tried to map this fruiting period QTL with short reads, we got a lot of noise. We didn't get any signal at all. We have no genetic control of this trait. When we go back and sequence with long reads and we map it again, we have this wonderful high effect QTL. And this is all sort of hot off the press, so the next part of going back in and using it in the breeding program is in process. But as you can see, all of the work and all of the effort and all of the investment into trying to develop these tools, with inadequate technology, we can't see it. But using the scaled long read approach, now we have a tool that can make a big impact for just one industry. And the third thing I want to share with you is from Alex Harkis's work. And Alex is really interested in the evolution of sex chromosomes. And I'm not going to butcher all of the wonderful biology that he would so elegantly say up here. But the fact of the matter is, the Y chromosome in the sexual determination region is actually a lot more diverse within plant species than we thought before. So if you're trying to understand how to select for males and females within these complex plant species, it becomes very difficult, and you have to understand the variation appropriately. And so he set out on a project with Zach Stansel at the USDA in Cornell, and they have started sequencing the entire germplasm collection of hemp with long reads. And just looking at sort of the early returns of how impactful that can be, what he's showing here is the pseudo-autosomal region, which reacts more or less as normal, and you can see the indels and duplications and inversions and the variation in the Y chromosomes within this large population. But what's really interesting to them is that in the SDR, where it should actually not have a lot of variation between individuals, they're still picking up a ton of variation that was never before accessible to them. And so there could be no insights. And I can speak out of school a little bit if I say that I've heard a lot of folks that work on hemp talk about the magic hemp genome. And this is probably why. Because they had no idea how variable the Y chromosome actually was. And so they just thought it was magic. It just didn't behave like we would expect. And so this is another way that you can show real impact in a species that has an impact in the way that people can make a change. And so to end, and I'm right on time, which is wonderful, we are offering and starting up special pilot pricing to get going with folks that can understand how they might utilize scaled long-read sequencing for their neck of the woods, for the organisms that they care about. And you can just imagine the examples that I showed, how might that drive innovation in your space? And we're excited about the power of scale and bringing the accessibility of this really profound innovation to those folks that want to utilize it. So with that, thank you very much, and I'll take some questions.